Hey everybody, my name is Christian Lugo. Nice to meet you all. Thanks for selecting my video to watch. Just a little bit about me. My name is Christian Lugo, as I said, and I work here at Chevron as a chemist. I refine jet fuel using catalysts that we develop here in-house, and I'm here to give you guys today a presentation on the organism that causes the condition called Tinea Versicolor. So as you can see here, the Tinea Versicolor is a dimorphic fungus. This is a yeast. So typical yeasts like Saccharomyces and Pitrosporidiums, any spore-producing microorganism. This is what the condition of the Tinea Versicolor is caused by. It does cause an infection. It is considered a parasite because of the way that it requires a host organism to exist and thrive and reproduce on. And it overcomes our innate immunity through its evolved tactics of being able to sense self, being able to trick our immune system from not recognizing it, and it overcomes our adaptive and cellular and humoral and innate immunities through its evolved tactics, especially concerning the chitin and the lipopolysaccharides that are on the membranes of the cell walls of the fungi. So we can treat it. It's good to know that we can treat it, and I'll talk to you later about how we can treat it. And I'll talk to you, essentially, this presentation is going to go over what the taxonomy of this organism is, because it's important to know the descendant and the phylogeny between this organism and other species that may or may not be related, in case you're considering the potential for zoonosis or for transmissibility. The taxonomy becomes part of an interrelatedness of species. Then we're also going to talk about how the species was discovered, and because of the discovery, the morphology and the description tactics that were used by biologists earlier on in like the late 1800s. And then we're going to talk about how to culture it and how its structure is related to its culture. And we're going to talk about how it overcomes our immunity through the exact chemical signatures that it releases, as well as our adaptive and our biological component that's involved with it. We're going to take a look at what it looks like and how it produces scales on our skin. And we are also going to talk about how to classify its pathogenesis through the cellular, the innate cellular, and the adaptive cellular processes. And lastly, we're going to talk about how to treat it. So if you want to join me on this little description, then an explanation, illustration, then I'll happily take you along. So here we have a microscopic cell, typical microscopic slide, and you can see here on this stain that this is a skin scale of a patient who presents with Tinea Versicolor. You can see the dimorphic fungus, so you can see two different shades of the same organism. They're not different organisms. That's why it's important to use this proper staining technique, like potassium hydroxide. And it shows you the germ rods, which you can kind of see right here. These are kind of the elongated hyphae or pseudo-hyphae of a fungus. And then the spore-producing cell, that's almost the spherical shaped fungi. This is the growing portion, not the reproductive portion. So Tinea Versicolor is caused by the organism called P. orbicularis. P stands for Pitrosporidium. So you can see the pitro meaning skin and sporidium meaning fungi. So these are a type of species that are skin-related yeast species and cause skin-related disorders. So we can talk about the taxonomy. The taxonomy is a Malaysia species, and that goes into why it was named like that. But the Pitrosporidium and Malaysia, just for future reference throughout this presentation, are synonymous with each other. They are lipid dependent, which goes into show you how they can kind of culture it on our skin. Because when you think, okay, how is there a typical, what nutrients are these organisms obtaining? And you have to consider they only have skin, they have air, and then they have our oils produced by our skin, and that is actually what they use as part of their respiration processes. So these fungi, these fungi are of the kingdom of fungus and phylum of Basidiomycota, our typical fungal species. The species and genus, as you go down into family in classes, it's just tends to be Malaysia, but the species is actually P. Ovale and P. obiculare, which is what we're going to talk about. And those have to do with the shape of the growing cell. One's more spherical and one's more orbicular. But together they're known as Amphiphora as a single species. These parasitic yeasts are different from other of the parasitic yeasts, even though they both are related to skin diseases, is because of their lipid solubility versus their lipid dependability. And the lipid dependent one is the one that causes the Tinea Versicolor, the P. obiculare, and it is characteristic of human host of potential, the ones different from animal skin conditions and different from different regions of the body, definitely. There are different species of the organisms that cause dandruff. There's different species of the organisms that cause psoriasis, that cause typical atopical dermatitis, and this one is called Tinea Versicolor. So Malaysia species, let's talk about that. How did it get its name? And so we can credit the name of the Malaysia, or however it's pronounced, molasses species, from the original founder, Louis Charles Molasses. This is a photograph of him. He was the person who actually classified and did a dichotomous index of the species of yeast that we're talking about here. So just a little bit of history. There's actually another person, there's a theory of independent theories of a rising science, and two different people at the same time, similar times, actually discovered the same species. But it is actually credited to Louis Charles because he was the first one to coin the term and coin and classify the species. They are now given most of the skin-related fungal species are actually all given under the genus Molasses. We can thank C.P. Robin for actually combining the two into a single category known as M. furfur. So we won't talk about it as M. furfur for the remainder of this presentation, but for us to know that it is, in fact, P. obiculare that we are going to be talking about. Even though it's related to P. ovale, it is still, both of them together, M. furfur. Louis Charles Molasses is a very credible scientist. He did a lot of work on hematology and dentistry and orthodontics, but we're going to give him his credit for what he did in mycology for classifying this species. So, okay, how did he do it? That's the question, right? Yeasts, the answer is simple. The yeasts are only cultured on a peptone glucose medium agar. That's the only way you can get a yeast to culture. You can typically do it in 5 days with about 7% carbon dioxide atmosphere in order to mimic a proper culturing condition for them to grow and to do proper cell counts, colony characterization, obtain cultures for staining, and the latter, right? So we can see that the culturing, that's how you typically do it. And as we looked earlier, we can see this is what a typical culture would look like. So this culture translates to this slide, translates to the key aspects of these yeast cultures, which are the dimorphic species of producing a budding spore, a budding germ rod, and a growing cell. And you can see that on this. You can see that on the slide, and that's the main structure and culture. It does have, like I said, with Tinea Versicolor, it does have other conditions like signatures and like the indoles that it produces, and how it kind of changes a different color, which we'll see later. Actually, I can show you really right now. This is the color change related to the respiration of the P. obiculare. But these are the main aspects that really talk and classify P. obiculare from other species: is the hyphae, the pseudohyphae, the germ rods, and the indoles and the chemicals that are produced. So I talked about, we're talking about culturing it, right? Typically, I'll just use this as an aside, typically cultures of this species aren't really necessary. It is very easy to determine from a trained medical observer that this is the species, this is what's happening. But if and when you do need to do a culture, you have to use a glucose yeast peptone agar extract, and like I said, the 7% CO2 for 5 days. But these indoles that we're talking about, right? This is one of the signatures produced by the organism. These organisms produce signatures that are related to their own genetic potential. That you can see here, these chemicals that are created, right? These are what we attribute to the pathogenesis and the fungal infection. This is what gives the species the ability to colonize. You can see here on this side, you have your Pitria chemicals, right? These are the chemicals that enable it to grow on the skin, right? And then you have your indoles here. These indoles are actually what prevent it from being recognized by our cellular immunity. It prevents the this is beautiful. This is a beautiful evolutionary process, right? It prevents anti-inflammatory processes from happening on and under the skin that it is actually colonizing. By doing so, these chemicals prevent a cytokine response, prevent inflammation, and actually prevents the host from being able to recognize that there's actually an infection taking place. These chemicals over here, these Malaysia, these indoles, these malices and indoles, and fungal chemical, anti-inflammatory, anti-infectious agents, or and not infectious. I'll save that word for later. Anti-inflammatory agents. These are the signature chemicals produced by the Malacia species to overcome our adaptive and primary immunity. These chemicals are actually what differentiate taxonomically between the different species of Malacia. This one right here, these carbazoles, these are actually as a result of the carboxylic acid digestions of the lipids, the fatty acids, right, that are produced by our body, and they can actually turn these into the carboxylic acid into a carbazole, and this is what actually causes the characteristic staining that you can see right here, the staining, the pigmentation, and the alteration of the pigmentation of the skin, right? So now we know that chemicals that are created by it, right? We're going to look a little bit into the pathogenesis, because it's able to, like I talked to you about, overcome our innate immunity through and create a pathogenesis response, right? So infection, this is what happens. This is the meat and bones of the microbiology of this organism, right? So let's consider this organism between us and our innate immunity and the species, right? The relationship of our body at the chemical and the biological, at the cellular level, focuses on 2 points, right? It focuses on the pathogen-associated molecular patterns. These are the signature patterns that are created by the infectious organism that, like the membranes, the germ rods, the hyphae, the products of the respiration, the products of the metabolic processes, the catabolic processes that occur inside the organism, right? These are called pathogen-associated molecular patterns. These are like polysaccharides, peptidoglycans that are unique to this pathogen and mimic natural peptidoglycans that we have in our body, so that they can overcome our detection in our cells by our IgG, by our C3, by our C4, by our helper T cells, by our macrophages. These do so by modifying its chitin and its beta-glucan on its membranes, right? However, it's not to say that it goes without a fight, right? Our side of the portion is the pattern recognition receptors. These are secreted by our immune system. These tend to be very small protein complexes, part of our major histocompatibility complex, like our IgG, right, and our MCHC, that are released by our cells, especially like our lymphocytes, right, to target antigens that it may recognize. The fungus aren't foolproof, right? When they're able to secrete these anti-infective therapies, right? These anti-infective chemicals that are released by the body, and they actually bind to a pathogen-associated molecular pattern. So these two actually bind together. That is what targets the beginning of our cytokine response, the beginning of our inflammatory response, but it often doesn't really make it. This is a minor disease, a minor pathology, but it does overcome, but it does able to be bound, right? They can bind together, and then it begins the recognition of the process of the adaptive immunity, right? So the let's say a pathogen-associated PAMP, right, binds to PRR, the adaptive immunity then begins to take place, and the generation of the T helper cells released by the B lymphocytes to bind through adaptive CD4 plasticity to create those immortalicized T cytotoxic T cells that are able to then bind, because of the pattern recognition receptor bound to the molecular pattern of the species, bind to the organism to help kind of signal through like a chemical signature to our TC cells that this species is foreign, and begin the process of our host immunity. That's why these Tinea Versicolor is actually resolved on its own very quickly, because our adaptive immunity took place. Our cells were able to recognize, our cells were able to stop the infection, and we're able to mediate it, right? So let's say that, and then it's been released, right? And then low levels of our atypical T cells, TC cells that are maintained in the serology, and that's how we can resist infection and further infection by exposure, right? That's talking about the pathogenesis, and then we can talk about the cellular physiology, right? Here you can see the macule. The macule is, like I said, that skin scale that we see when people present. This is a mole. Don't worry about that. This is we can see when people present Tinea Versicolor. Just to let you know, the Tinea Versicolor isn't reaching through all 5 layers of your skin. This is actually only a species that can only colonize on the stratum corneum. These are like the most upper layer, right? So the thin parts of their skin that people have. So you won't be seeing Tinea Versicolor on something like your foot, on your heel. You won't be seeing the Tinea Versicolor species on your scalp. That's actually a different species of Malassea. But on something like the upper arms, on the chest, on the extremities, anywhere here on this torso, is where you would see Tinea Versicolor. Some of the factors that affect the epidemiology, like I said, is the humidity. People in regions with higher water are definitely more prone to culturing some organisms like funguses. Tropical climates, definitely you can see yeast growing. You can see patients, unfortunately, with like hepatitis C and HIV and AIDS untreated. With lower immune systems, they aren't able to mount those sort of TH cell recognition to create the B cell and the cytotoxic T cells that we need to fight infections. Unfortunately, some peoples have different hereditary factors that might affect them. Some people have different lipid production levels. Some people have different genes for skins. Their oily skin, it tends to be that risk factor for somebody who might have Tinea Versicolor. It also affects the people of age more differently. So younger children and adolescents tend to be more infected with this parasite than adults. But notably, actually, hygiene has nothing to do with this. You can wash and wash and wash and wash and make sure your skin is dry, but it doesn't mean that you won't be able to be infected by this organism. So that's what happens, right? We talked about how you can culture it, how it gets through our body, right? The pathogenesis, how it goes over through our innate immunity and then through our adaptive immunity, and then we talk about how to treat it, right? So treatment and prevention. This has been known for quite a while, actually, since 1996, 1983. People have known that I had a little visual for you. So you can actually play it. So people can know that the Tinea Versicolor is can either be topical or versus systemic, right? So what does that mean? Topical infection is something like very small, very patchy, like a couple areas, right? Can be treated with a little bit of shampoo, a little bit of cream. Versus a systemic infection is something like widespread, right? Definitely on the hands and legs and the groin and the neck. Those are something that a physician would consider. Okay, this is not a acute localized infection. This is actually a systemic chronic infection. If it keeps coming back, they actually recommend not to use the topical shampoos and creams, but actually to take it orally. That's actually the most. It's rare, but that's the logic process on how a physician would approach this. So how does it work? Oh, I moved it. That's my fault. How does it work? So it's similar to penicillin. We'll talk about ketoconazole. This is actually the main chemical that's the treatment that is the pharmaceutical that is actually used for treating the fungal infections. How does it work, right? The similar to penicillin, ketoconazole works by absorption of the chemical into the fungal cell, and that chemical actually disrupts the synthesis of, let's see, I have the exact word for here, the synthesis of its membranes. Is the synthesis of ergosterol, which is used to build its membrane. So when you have a component like ketoconazole inside the fungal species, it cannot build its cell walls, it cannot do mitosis, and eventually dies. Right? This leads to cellular death. Your infection is cleared, and it does it exactly in a similar manner to it is a entry mechanism related pharmaceutical that kills Tinea Versicolor, or kills the P. orbicularis species and prevents and resolves Tinea Versicolor through synthesis blocking, just similar to so many other antibiotics that we have. It's relatively quick. It often doesn't need to be going into the systemic process, but like I said, this is a typical treatment for Tinea Versicolor, and then the prevention of it in the future is very simple, right? The individual just has to go through the adaptive immunity here and prevent further colonization efforts through our T cell recognition, our T helper cell, and our our B cell, atypical B cells that are released as a result of the infection. And our adaptive immunity is what prevents the further infections from then on. And then we can go into systemic infections, but that's not what we're talking about here. So, yeah, I just think that throughout the process, right, we talked about how it looks like on the cell, who invented it, how to culture it, what signature is actually unique to this species versus other species. You can actually detect it too if you needed to. I talked a little bit earlier about it, but the Tinea Versicolor can be observed and diagnosed by the eye. It doesn't often need to be cultured, like I said. But if you wanted to do a quick test, you could do a potassium hydroxide test on a stain. And if you were to do that test, you would see these little hyphae here, and that is what shows you that it's a fungus infection. And then you can begin the antifungal treatment that we know and are currently using. So you have the infection, how we prevent, how it actually infects, where it infects, why it infects, and the sort of the epidemiology of it, who's susceptible, why are they susceptible, how is it transmitted. It's actually very, very, very low transmission. Like it's not spread from person to person. That's another key point. But yeah, I just wanted to thank you guys for listening to my little presentation. I know it's not as formal as if it was me standing, but this is the presentation on this Tinea Versicolor dimorphic fungal species that cause some spotting on people. So thank you, and I hope you guys have a great day.